and welcome to the third event in our 2022 speaker series, Surprising Science, Borrowed Ideas Leading to Unimagined Consequences. I would like to acknowledge and pay respect to the traditional owners of the land on which I am this evening, the Ngunnawal people. It is upon their ancestral lands that the Australian Academy of Science is built. As we share our own knowledge, teaching, learning, and research practices, may we also pay respect to the knowledge embedded forever within the Aboriginal custodianship of this country. I am Nanda Dasgupta. I am a fellow of this academy and one of the conveners of this series. We thank you very much for coming on this cold Canberra night, and as a compensation, one of our speakers is actually up at 3:45 a.m. in Canada because she happens to be in Toronto. Anyway, joining me to convene this series is Professor Drew Evans from the University of South Australia. Hello, Drew, and I'll hand it to you, Drew.
Thank you, Nanda. It's an absolute pleasure to be working with you on this series, and thank you very much for coming along this evening. I think it's safe to say that we've got so many creative ideas coming through this year that this is quite an eventful speaking series. So today and this evening, I'm proudly joining you from Kaurna country, and I too pay my respects to elders past and present. And thank you all for joining us for our latest event in this series. So tonight we will hear about fingerprints in our oceans. Dr Indrani Mukherjee and Dr Zoë Doubleday will no doubt surprise you with their science, and probably even more surprising will be the connection between their science. Now, just a few things before I hand over to our speakers. So that our audience can engage in tonight's event, we'd like to just talk through a couple of ways you can do that. So if you'd like to join the conversation on Twitter through social media, you can do so by using the hashtag #SurprisingScience. For those of our audience who are joining us online this evening, you'll be able to submit questions for our speakers using the QR code on screen. And for those of us joining us live tonight in the Shine Dome in warm, sunny Canberra, as a former Canberran, I know winters well, you'll be able to ask questions during the Q&A session by using the microphone within the Shine Dome. And with that, let's now hear from our first speaker, Dr Indrani Mukherjee. Indrani is a researcher and lecturer in earth sciences. Her research questions key concepts and explores links between early Earth evolution, the origin of complex life, and formation of precious mineral deposits. Some might say Indrani is going to take us back in time. In fact, we might just check in with her now. As we were conversing earlier, she's speaking to us from the past. So Indrani, how are you?
I'm doing really well, thank you, Drew. And it's wonderful to be part of this. I can confirm that it's lovely and warm in Toronto right now, and we'll just forget about the time for now. Maybe I'll think about it later after my talk.
Fantastic. Well, welcome, and a really, really big thank you for joining us all the way from the other side of the planet. So the floor is yours, Indrani.
A pleasure to be here. Before I give my presentation, I would like to acknowledge that this work was conducted in Lutruwita, Tasmania, the land of the Muwinina people, one of the oldest continuing cultures in the world. So today I will be talking to you about pyrite fingerprints in ancient oceans. But let's have a look at this first. What comes to your mind when you look at something like that? And I refer to the image on your left specifically. Just a blob, or rather probably nothing, just a blob of blackness. But what if I were to tell you that that there represents one of the beginnings of complexity of life on Earth? An event, a biological event, so remarkable that you and I and the millions and millions of species on our planet, we owe it to that particular event. And why is that? You know, you and I are composed of 34 to 37 trillion of what we call the complex eukaryotic cell. And this image right there marks one of the beginnings of that complexity that we had on Earth. So just to give you another idea, that this single-celled image that you see on your left here, the single-celled organism, something happened, something remarkable happened, and that transitioned into a very complex eukaryotic cell that you and I and a lot of complex organisms, macroscopic organisms today are made of. In other words, we would be safe to say that without this transition, there would be nothing on Earth today except bacteria. So that's how remarkable this transition is from a very simple, although very sophisticated organism, which is the prokaryotic cell, to this eukaryotic complex cell. Now, I'm not going to pretend I'm, know-all biologist, and so right up front, I want to be honest with you. Trying to understand important biological events in our Earth's history requires the knowledge of chemists, biologists, and geologists. No single discipline, no single scientific discipline alone can solve this conundrum. And so evolution of complex life on Earth, again, is a very complex and dynamic subject. There are questions as to when this transition occurred, how this transition occurred, and of course, the most important question, why did it occur? If a single-celled organism is perfectly living on its own independently, why would it convert into something so complex? And so these are some of the questions that surround this dynamic topic, and I will go through each one of you, but I will let you know right at the start that it's the why that geology can attempt to answer. But before that, let me just go through what evolution of life on Earth may look like. Very simple schematic representation of what evolution of life it looks like on Earth. And so we obviously start with very simple microscopic organisms, and then things get bigger and more complex. And then, of course, we have, Freddie Mercury here representing, honestly, he is the benchmark of complexity, if you ask me. But essentially, the idea is that you start off with very simple microorganisms, and then that gets more and more complex. What if I were to put this into perspective with the Earth's timeline? And so in this slide here, this is what I'm trying to show. All the macroscopic organisms, all that we see is just concentrated in that very tiny period between just over 500 million years ago and beyond. And so prior to that, we had this key period of eukaryogenesis. I know it's a big word, but all I mean is that genesis of that first complex cell. It was of utmost importance that that event had to occur for us to be able to see the complexity that we do on Earth today. And so this particular time here basically represents a very key period, and you've got microfossil evidence of it ubiquitously present in the rock record, telling us that this particular time was incredibly important for the genesis of the complex cell. So we know roughly about the timing, right? And so then let's move on to how. So on the image, on your left here is, I've named it predation or hostile. Did two organisms interact in a hostile manner? Because we do know that one larger cell or one cell engulfed the other. Now, whether it was a hostile interaction, meaning predatory in nature, we don't know, but some researchers believe that it was predation. Whereas on our right here, we've got endosymbiosis, which is more of a friendly sort of interaction where two organisms combine or join in to for mutual benefit. And both of these examples are from modern day today, by the way. This is not from back in time, but this is just to give you an example of that we see these sort of interactions in the modern oceans today. And so it could have been that two organisms either interacted in a hostile manner or in a friendly manner. Regardless, the result was fantastic. But that's again debated, right? Whether which pathway the organisms take to combine together, and in course of time, these sort of combinations of having these different cell organisms coming within a larger cell and then forming the complex cell. Now, how this introduction took place is obviously debated, but it could be both predation or endosymbiosis, so friendly. And here's where geology features. Why? What sort of geochemical environment did Earth present to the then prevailing organisms for those interactions to occur? Because organisms respond to the elements that have been made available to them through course of time. They will react to abundance, they will react to deficiency, they will react to critical balances of these particular elements. And so all these elements that are required, they're absolutely critical and bioessential. The introduction of these elements into our oceans depend on these broad-scale dynamic geological processes. For instance, tectonic settings. Are we having enhanced plate tectonic movements, or are we having suppressed, meaning are we having more mountain building, which will cause more erosion and leading to more nutrient supply in the oceans? Or were the continents rather more stable with suppressed erosion rates and therefore a rather mild supply of nutrients into the oceans? What was the atmospheric redox like? Because you can have a lot of erosion, but you need the right type of rock that needs to be weathered for the eroded material for them to be transferred into our oceans. So there are these really broad geological factors that basically determine what we introduce or what the Earth introduces into our oceans. And that's because the chemical environment that we provide, or the Earth provides to the organisms, is absolutely key, because every single life form on Earth today depends on these elements. So that begs the question, what other elements are essential for life? And of course, the common assumption is oxygen, but I've highlighted some elements in red and blue here. And that's because it's a very human way of looking at things. Oh, we need oxygen, so maybe everything on Earth needs oxygen. And that's not true. Even today, 50% of the world's oxygen is actually produced by our oceans, by our phytoplanktons in our oceans. So they don't require oxygen. They in fact produce the oxygen. And so focusing on just one element is not the right thing. We need to look at a suite of them, and particularly the one that come in short supply, and they're known as trace elements. And so the ones highlighted in red here. So my research uses geochemistry or geochemical techniques to understand how trace elements through time, or bioessential trace elements through time, have varied, and what were they particularly doing in that key period, to see if that can provide me some insights into why that transition from a very simple to a very complex cell occurred. And again, this is reiterating what I said earlier about oxygen requirements of the earliest animals. It is a common assumption that oxygen is very important, and it is. I'm not saying it's not. But I'm just saying when we're looking at things that far back in time, we also have to keep in mind their requirements far back in time. Oxygen is a requirement now in an oxygenated atmosphere with rather large macroorganisms that require oxygen in their cellular functions. But back then, things were rather small and they could have done well without oxygen, as also demonstrated in this study here. And so significance of trace elements, apart from the fact that of course Superman himself needs trace elements to operate, really it is incredible how important these trace elements are, not just for microorganisms but for every single life form on Earth. So I've listed 3 basic bullet points here which you may read through, but to give you in a sense, this is the sort of complexity. It's a very simple, beautiful, this is when the wine tonight comes handy, everyone. A very simple diagram. I'm sure you can all understand it. But see, this is what I mean. The temperatures and pressures that can sustain life are, however, not suitable for the various chemical reactions that keeps life going. So these metabolic pathways or chemical reactions are all linked. And these reactions will be incredibly sluggish, sometimes won't even go forward under current temperature and pressure. And so they need to be sped up 1,000 times. And they can do that through trace elements within enzymes. So trace elements are incredibly important to not just sustain and grow life, but the basic survival. And so I was interested in understanding, well, what was the trace element availability like in that key period for complex cell genesis? That will provide me some insight into maybe the chemical world had something to do with the biological. And so I use pyrite fingerprinting to understand how the whole trace element availability through time works. What is pyrite? I'm sure you've heard of it for all sorts of things, but it's essentially an iron sulfide, and it comes in all shapes and sizes in our rock record. I'm using pyrite fingerprinting to understand or answer one of science's biggest question: what drove biological transition of a simple cell to a complex cell? But I will have you know that pyrite fingerprinting, depending on the geological environment, has multiple applications. Whether it's astrobiology, whether it's mineral exploration, it really is that really super useful mineral whose chemistry can be used to understand all sorts of things. But for this presentation, I will focus on how I've used pyrite to understand what the trace element availability was like in that specifically key period. And so how does it work? So I touched upon this briefly earlier on, but essentially the premise is that enhanced erosion owing to enhanced plate tectonic activity or mountain building leads to higher rates of erosion. And when you combine that with oxidative weathering on continents owing to higher levels of oxygen in the atmosphere, a combination of this weathering and erosion leads to an increased supply of bioessential critical elements through the riverine flux into our oceans. Now, once this a suite of bioessential elements are introduced in our marine system, they are obviously then being made available. A common notion is something that's available, it's good, something that's not available must be deficient. But I think organisms are much more sophisticated and complicated than that. Sometimes higher abundance of an element will trigger a very different response to the ones that are in short quantities. But in a sense, these elements are made available through weathering and erosion on land, and once they're taken up by these organisms, they utilize it for various purposes. But when these organisms die, they deposit on the sea floor with the muds, and these muds are usually organic matter rich owing to the organic matter deposition. These organic matter rich muds eventually form what we call black shales through time. So when these muds are lithified in the rock cycle, we produce black shales. But the beautiful part of this is that when this mud forms, what we also form is pyrite. And when the pyrite is forming, the trace elements that were once taken up by the organisms are released when they deposit, and these trace elements are efficiently absorbed by the pyrite forming in the muds. So in a sense, by understanding the trace element concentration of marine pyrite in marine black shales or black muds, we are able to measure or at least have an idea of what trace elements might have been available during the time of its deposition or formation. So looking at the trace element chemistry of the mineral pyrite, and specifically sedimentary pyrite that form in black muds, we are able to track the availability of trace elements through time. And so what do things look like in real life? So if I were to show you, this is what a typical black shale sequence would look like in the geological rock record. And so going back to that rock record in geological timescales, this is a black shale sequence, and what I do, or what my research does, is traverse across these scales. So, you know, from meters to micron level, and it's just beautiful. So just demonstrating that, if you had a close-up, you can see the individual pyrites forming in these, well, will have formed a long time ago, but in the black shales. And then if you have further close-up and you look at it under the microscope, you start seeing these beautiful framboids, we call them, of pyrites, and a close-up of one of the pyrite framboids here. And so essentially I measure the trace element concentration of this particular framboid here using laser ablation mass spectrometry. And yeah, so essentially using that geochemical information, I'm able to construct how these elements and bioessential elements have differed in or have basically tracked through time. And here's a graph, again, nothing to be scared of. I will go through it very briefly, but just reminding you of the key period that I referred to, and so I'm showing you that particular period here. On your x-axis you've got time, and we're specifically focusing on that time marked in a dashed rectangle here, and on your y-axis you've got nutrient availability. So essentially it just gives you some ideas. So lots and lots of analysis, but you are able to construct a nutrient availability trend through time. And yes, it's got lots and lots of ups and downs, but really what I'm trying to show you is that there was obviously a very nutrient scarce period, which is below the black dashed line there. And then obviously the nutrient availability recovered soon after 1500 million years ago. So within that key period that I've been referring to this entire time, at the start of that key period, you seem to have had a nutrient scarce period. Now, this is all relative. And then around 1500, the nutrients recovered. What does this mean? And how does this stack up with the rock record? It is that nutrient scarce period when we see biological innovation. And when nutrients availability recovers, we see diversification. And the way I've used to explain this is that that nutrient scarce period may have imposed an environmental pressure or stress on the then prevailing organisms. I personally think this nutrient scarce period is the very reason we have the complex cell. Because you have to think of this. Why would two cells interact in a friendly or a hostile manner if they are completely capable of living independently? The reason why we could expect these interactions to occur is because you suddenly change the geochemical environment, and that low nutrient period probably marks as the first striking variability in the chemical environment in Earth's history. So there was a very striking change, and organisms had to adapt to that change. And having something scarce will bring organisms either to fight or in a friendly, depending on how they want to interact. But that essentially that nutrient low period or nutrient scarce period, in my opinion, will have been a very important part in our biological evolution history, just prompting organisms to take those steps, whether predation or endosymbiosis. And of course, when nutrient availability recovers, that's when organisms will have already evolved under those stressful conditions and will have better traits, and so that will help them with diversification. And so just reiterating, nutrient scarcity will have caused stress, and that would have been a major trigger for biological evolution. And nutrient availability with recovery will have been a huge trigger for diversification. And so this is how I've used geochemistry of our oceans, and particularly the pyrite fingerprinting in our ancient oceans, to answer how, why we have the origin of the first complex cell. And to conclude, why waste a slide writing thank you when I can personally thank you, but I will take this opportunity to wish you all happy Pride Month and show you some graphics by Indian rainbow community artists. Thank you.
Thank you very much, Indrani. That was absolutely fascinating to hear how geochemistry and applying geochemistry today with the tools and techniques we use can actually tell us a lot about what happened a very, very long time ago. So not just from us sitting here on in Australia, sort of going back about 12 hours to where you are, being able to go back hundreds and thousands of millions of years to really make some deep insights into life as we know it today. So very much thank you for that, Indrani. A fascinating talk.
Thank you for the opportunity.
And next, I'd like to take the opportunity to introduce our second speaker, Dr. Zoë Doubleday. So Zoë is an award-winning marine ecologist and an ARC Future Fellow at the University of South Australia. She uses geochemical tools to unlock the biological histories and geographical provenance of marine animals, as well as deep solutions for the seafood and fishing industry. To hear more, please join me in welcoming Zoë.
Thanks, Drew. It's wonderful to be here in Canberra, and I got my first time on a plane for a long time. I even had to read the instructions on how to board a flight again. It's been so long, so it's so great to be here and do something face-to-face. So before I start, the research I'm going to talk about was done on Kaurna country, and I'd like to pay my respects to the traditional custodians of Kaurna country and also Kaurna Sea country and their incredible environmental stewardship. So I'm going to be talking about ocean fingerprints, but for a very different application. And it's like almost creating what I call a GPS for marine animals. Can we position them on the globe and find out where they come from? So I'm a marine biologist. I use geochemistry, but I feel I'm very much a pretend geochemist. I work with amazing geochemists. I'm not one myself. And this is a little guy I work on a lot. This is a hatchling we had in our lab about a month ago. He's about a month old. And one aspect of marine animals that I study is their movement and their provenance. Where do they come from? Where do they go to? And this sort of data for ecologists like myself is really useful for all types of practical applications. One is understanding movement and provenance of animals, and this could be on land or sea, can inform conservation planning. For instance, you might find that these mangroves pictured here are optimal nursery grounds for particular species, which might be threatened or it might be commercially important. You might then, those animals, once they grow up, they might move to breeding or foraging grounds, which is a different sort of habitat. So it can inform what habitats we need to protect most, and it can inform, say, where we put a marine protected area. One other aspect of understanding how animals move in the ocean is in fisheries management, and this is an area I've worked on, in terms of one of the key aspects a fisheries manager wants to know, whether so they can manage a fishery sustainably, is what unit, spatial scale they should manage that fishery. For instance, say you have a commercial stock population of snapper across the southern Australia. That could be just one homogenous population of individuals all intermixing, or can be composed of discrete little subpopulations where there's not much interchange between those units. If they're made out of discrete subpopulations, you want to manage those units as discrete subpopulations because you could easily cause localised extinction because there's not replenishment for coming elsewhere. So this is one of the key reasons why we collect this data. So you can also look at the movement using the same techniques of animal products like seafood rather than living animals, and this can help us detect things like seafood fraud. So unfortunately, consumers and businesses can be intentionally deceived about where seafood comes from for profit. And that allows illegal fishing and unsustainable fishing practices to flourish. So if we can help detect that fraud, we can help authorities, to help stamp it out. So one of the methods that I'm an expert in is using chemical fingerprinting to track where animals move in the ocean. So this is looking at elements in the tissues of marine animals which can reflect their living environment. It could be physical aspects such as temperature and salinity, or even the chemical element makeup of their diet. So we can use these tags or fingerprints to track where these animals move, but also track how the environment changes around them. So chemical fingerprinting is fantastic. We've used it successfully for many useful applications, and other methods as well, like DNA fingerprinting and physically putting tags on animals to track where they move from, is also very successful. But generally they're developed for one species or industry or taxonomic group at a time. So when we do chemical fingerprinting, say we get barramundi from location A and location B, we analyse a suite of elements in the tissues of those animals and see if we can use it as a fingerprint to track where they're moving to. But my research is focused to see if we can develop a more universal system. There's so many animals out there in the marine ocean, it's inaccessible, it's vast, it's diverse, and there's a lot of species that just go under the carpet. And so we can monitor a greater spectrum of marine life with fewer resources. So I'm just going to give a bit of a background about chemical fingerprinting and how I'm using it to develop a more universal system to track marine animals. So we love using shells and bones of marine animals, and this is because once the chemical's in that bone, it's locked in there forever, it doesn't change. You can analyse chemical elements in muscle tissue, but they can change depending on how the fish is feeling that day, how old it is, even post-mortem effects. So we can analyse archives of these bones that are decades old, centuries old, and we can still analyse the chemicals in there and they remain unchanged. So here I'm holding a beautiful fish ear bone or otolith from a Murray cod, which is an iconic freshwater species that we have. Marine species have the same structures, marine fish. And when you cut these structures open, you get this beautiful banding. So this is a cross-section of a black brim otolith. So the core, the dark core you see here, represents the birth of the fish. And this edge here represents the death of the fish. And each band represents a year. So it's like a tree. We can read it like tree rings, or how dendrochronologists understand trees. And we can use tools like laser ablation, mass spectrometry, which Indrani mentioned, and we can effectively use a laser to blast this material along a transect here and analyse the trace elements within that transect, and we can track it over time across the course of the lifetime of the animal and get all sorts of information. So shells and bones, most marine animals have them. So we've got these beautiful gastropod molluscs that I collected from a beach combing expedition there. And even the most surprising animals have bones, so I work a lot on octopus. Now, if you squint really closely, I promise there's a white speck on the tip of my thumb there. And that is actually the ear bone, or what we call statolith, of an octopus. Now, octopus are famous for being boneless and getting into all sorts of different cracks, but they do have these little bones underneath their eyes, which they use as sensory organs. And we can actually analyse those bones and track the provenance and movement of octopus. So this got me thinking. So I've worked on all these different structures throughout my career, using a species-specific approach, so optimising chemical fingerprinting for one species at a time. But thinking all these shells and bones from such a divergent group of animals, so we've got an octopus and fish, for instance, having these shells and bones, and they're all made of calcium carbonate, the mineral. So we're more closely related to fish than octopus and fish are, because octopus are invertebrates. So we've got these really different animals that have calcium carbonate structures. This has got me thinking as, can we do chemical fingerprints that are more relevant to more animals? So again, as you see, you've already seen the periodic table. So with standard fingerprinting, we analyse all sorts of trace elements within the bones and shells of marine animals. But I am particularly interested in oxygen and isotopes of oxygen here. And isotopes are just different versions of the chemical element. It's usually expressed as a ratio, so you ratio version A against version B, and it can tell a lot about how the environment's changing and the environment that the animal's living in. So this is when I use decades-old research from geochemistry and paleoclimatology, and who want to predict the temperature environment of years past, because they've developed these really precise relationships between the oxygen isotope value in calcium carbonate structures and seawater temperature. Now, we've got brilliant data on seawater temperature, but I don't want to use it to reconstruct past temperature environments, like say paleoclimatologist wants to do. I want to use it to see if we can geolocate animals on the globe, like use it to develop a GPS for animals. So in theory, because all these animals have calcium carbonate bones, in theory, an octopus, a fish, and an abalone living in the same body of water should have the same oxygen isotope value in their tissues. So this is what I'm starting to explore, and this graph here is a bit of a product of COVID, because when COVID started, we weren't able to travel to collect our samples and we weren't able to analyse them. So what we did instead is compiled a global database of all published values of oxygen isotope values found in the calcium carbonate tissues of marine animals. So we've got the key ones. We've got fish, we've got cephalopods, my favourite, which are cuttlefish, squid and octopus, and we've got shellfish, which are like your bivalve mussels, bivalves and gastropods, things like abalone, mussels, clams, scallops. So what you've got, I'll just orientate yourself on this graph here, is we've got here, this is the equator. This part of the graph represents the northern hemisphere. This represents the south. And particularly for the southern hemisphere, you've got this beautiful relationship with latitude. Now, I got incredibly excited by this because as a bioecologist, we deal with all sorts of noisy data, even if you get chemistry data from different individuals of the same species living in the same region, we can get all sorts of variability. And to see this was quite a messy data set. This data spans 25 years of published data. So we've already probably got influence of climate change as the ocean warms. We've got changes in instrumentation and the precision of those instruments. But even with that, you've got a group of animals analysed over 25 years living in, say, southeast Australia, very different animals on the phylogenetic tree, and they've got this same value, so that sort of blew my mind. So because of all the work done by geochemists and paleoclimatologists about this very predictable relationship between oxygen isotopes and seawater temperature, we can build a model, a map. We can predict what the values might be in those animals. So this is just this is not real data. This is what we predict will be in the ocean. So in the bones of the animals. So for instance, if we got this group of animals here living in the Indian Ocean, we'd anticipate that their their oxygen isotope value in their bones would be about -2. And again, in the Southern Ocean, we'd expect a value like here. So you've seen how we might develop a more universal system to track marine animals. So can we use this to create like a GPS? That's what I've been thinking. So we actually tested this using our published values, because we weren't going out and measuring our own values. So using that database I mentioned earlier, we compared whether we could geolocate the samples back to their known locations, just purely based on their oxygen isotope values. And we compared it between tropical Asia and particularly Southern Australia. And we did that because this study was focused on seafood. In Australia, 70% of our seafood we eat is imported, and most of it comes from the Southeast Asian region, so that's what we were focusing on. But to also play devil's advocate, we compared temperate, cooler water Asian waters with cooler water Australian waters. And then what we found was we could classify 75% of our samples back to site between the tropics and cooler water Australia down here, and a lower value, which we expected, because as I showed before, the latitudes have a similar value. But we got some feedback from a scientist, and they said, you know, some fish, only a small number, some species are actually warm-blooded, and they're like, oh, that's going to muck everything up. So most of the marine animals, you know, marine mammals are an exception. They're warm-blooded like us. They maintain a constant internal heat. Even if it's freezing outside, you might not know it, but you still have a same body temperature of 36, 37 degrees. Most marine animals are cold-blooded, so their or what we call ectothermic, so their internal temperature matches that of the ocean. So that all works well. But as soon as you get something that's warm-blooded, their internal temperature doesn't match that of the ocean. So one of the most famous groups of fish that are warm-blooded, what they call partially warm-blooded, are tuna. And we all eat tin tuna. They're a super important part of our seafood supply. So when we moved tuna from this study, we found that 90% of our animals could be correctly classified back to the locations based on that isotope value, which was just an interesting side point. So you might see a problem here. We sort of got broad latitude covered in broad chunks, but animals live on the same latitude. Obviously, the Earth is mirrored, and you know, 40 degrees north is similar to 40 degrees south. So how can we get longitude? Well, longitude, like, you know, maritime navigation, you know, latitude was easily solved, but longitude was much harder. You know, it's sort of a human construct. At least with latitude, it's defined by these nice environmental gradients which influence the ocean fingerprints. So for universal chemical fingerprinting, I'm looking at how to define longitude and improve the spatial resolution of this latitudinal marker that we've got. So developing different markers that have similar properties to oxygen isotopes. So one, we need the marker to be variable in the ocean. So on land, we can use things like strontium isotopes, but they're homogeneous in the ocean, so they're no good. We also need something that is not influenced by the physiology of the animal. So a lot of isotope ecologists use things like carbon and nitrogen isotopes to understand how the ecology of animals, but they're influenced by all sorts of metabolism, what level they are in the food chain, so we can't use those. So there's a different isotope that we're looking into called neodymium isotopes, and it's little known. Some Japanese researchers have measured it in animal tissues. But why it's so special is it's influenced by the underlying geology, the bedrock. So we can effectively create finer resolution of this universal fingerprinting. So I sort of see my job now is seeing the oceans like this paddock here. It's a series of different chemical fingerprints. And now I want to define where those boundaries are on useful spatial scales. Broad latitudinal gradients can be useful for a certain amount of things, like tracking seafood between Southeast Asia and Southern Australia. But we need greater spatial resolution. So where those boundaries are, and also what colours those paddocks are. If we've got dark green and dark brown, we can easily tell where those animals come from. But if you've got lighter shades in between, that can be more challenging. So that's the finish of my talk today. And yes, thank you for being a fantastic audience.
Thank you, Zoe. So good to hear about your work. And as a nuclear physicist, I was so impressed that you had the latest periodic table up to 118 elements marked correctly. So, anyway, I would like to invite questions from the audience here to both our speakers. So please, if you could come up to the microphone here, that would, for the audience in here. And for the online audience, if you could submit your question using the QR code, and Drew will look after all those questions. Yeah, Drew? I think that's how it worked best. Okay, so where shall we start? Oh, there are already 5 questions out there. Okay, Drew, you win. Come on, audience here. We have to win as well. So, Drew, you start.
Well, thank you, online audience. So to begin with, this is a question for Indrani. So in your presentation, you showed us some beautiful timelines of evolution, and on there you marked 2 particular events, and I think it's piqued the interest of quite a few people. Can you describe what the Great Oxidation Event is?
So the Great Oxidation Event, again, that someone there has obviously touched upon, probably, again, geochemists, in the geochemist worlds of controversies. So the Great Oxidation Event is a period in Earth's history. Now that's the controversy, whether it's this one narrow period or it occurred over an extended period of time. But regardless, it is a period in Earth's history where you had high levels of oxygen in the atmosphere. So, and that's relative to present atmospheric levels. So currently it's 21%. But Earth's atmosphere hasn't always had very high levels of oxygen. And so we've had to build up. And so some researchers believe that it was a sharp event at 2.33 billion years ago, which is the first Great Oxidation Event. And then things went downhill from there or remained constant. It's again a huge controversy. And then there was another significant rise in oxygen in the Earth's atmosphere. And then after that, oxygen has gone really high up, much higher than present levels, and dropped down to much lower levels as well. So it's fluctuated, but back in time, the time period I've been looking at, those 2 events are the periods when we had a sufficient oxygen levels in the atmosphere. We could spend a whole day arguing about when we should place these events, but no, that was a good question.
Indrani, we can carry on here for a few hours. You will be sleeping. Okay, Drew, you carry on, and I'm looking at people here, hoping that someone will come up. Okay, Drew.
Excellent. Well, this is a question for Zoe. How do you find octopus bones? So is it something that's easily recognisable, say in the sand on the sea floor?
No. You'll never see them in the sand. Can you hear me, Drew? Is that?
Yes.
Yeah, great. So you can get your eye in. So once you've dissected a few hundred octopus, you can do it like within seconds, but it can take a while to get there. They can be easier than fish, even though fish have, bones like this big. They have a skull because they're vertebrate, so you've got to sort of get through to that hard skeletal structure, which can be quite violent. With octopus, they're just soft-bodied, so you just get like a scalpel and you can just slice a little bit of the skin, and then there'll be 2 fluid-filled sacs called statocysts, And then and you'll see them, and if you just cut into them, the little ear bones will be floating around in those sacs because the octopus use those as sensory organs. You know, they float around, they use them for hearing and balance. But they are tiny, so often use tiny paintbrushes to handle them.
Okay, Drew.
All right.
Now the pressure on this audience is increasing. We have to have that 2 versus 0 at the moment. Okay, Drew, carry on.
Well, so now back to you, Indrani, and maybe moving off Earth for a little bit. So a question here is, given that a recent space probe has found an asteroid to have many kinds of amino acids present, what do you consider the likelihood of life of any type existing within the universe?
Big question.
That there is the most amazing research problem ever, and that's something I'm going to embark on. Actually, I've already started working to research similar to that. So looking at the rock record where the actual organism is completely replaced by another mineral. And so how do you distinguish between, for instance, I showed you pyrite. We see evidence of that on Earth. We call it exceptional preservation, where an entire, and you know, Zoe showed us some shells and bones as well. And so quite often, depending if the geological environment is right, it will be completely replaced by the mineral with the organism's shell structure absolutely intact. But then you also get normal pyrite in the rock as well. Can you distinguish between which one is biological and which one isn't? And so that's something that I've been working on lately as well. But the problem is, I don't know if it's a good problem to have, but for us, n equals 1. We don't have any other counterparts. There is a huge assumption that everything outside of Earth is going to be Earth-like. They may be. It's reasonable to suppose those sort of assumptions for Mars because they've had similar geological history, etc., etc. But to think we don't even understand Earth that well. And so to understand early Earth as well as possible is obviously key. But then you just have to pray, God only knows who to, that basically what we understand on Earth can be applied outside. And so if that's the case, then yeah, we should be able to detect these sort of remnants, what I would say, of organic matter, because similar processes may actually have occurred on Earth in the past. And so to scan rocks that are really, you know, we're talking billions of years old. And, you know, there are billions of years old black shales. The reason it's black is because the organic matter within those rocks have been preserved. Can we gain information from the different types of black shales preserved billions of years ago on Earth to then provide insight? So yeah, it's absolutely fascinating to even think about what can be done with the knowledge that is generated here.
Okay, audience here. No, maybe I'll pretend to be an audience. I was sitting there. So, Indrani, so when you talk of black shales, so that single cell organism that you're talking about, does that information also come from black shales or shales?
Some, yeah. So a lot of, so when I say black shales, and I'll repeat what I just said, it's because it's organic. So you've got normal shale as well, you've got grey shale, you've got all sorts of shales. But the black shales is black because of the presence of organic matter. And now very, sophisticated organic geochemistry, and in fact there's a brilliant organic geochemistry department right at ANU where they can actually assess what is the source of the organic matter in the shale, whether that organic matter was derived from a single-celled organism community or whether it was derived from a eukaryotic community or whether it was mixed, etc., etc. So it differs, but ultimately, you know, they don't discriminate like us, organisms, microorganisms. They all deposit in the shale when they die. And so there are ways of organic geochemists definitely do that sort of work all the time, where they look at certain organic compounds within the black shales and specifically focus on the organic matter and say, well, okay, this was derived from a single-celled microorganism community, or vice versa.
Okay, Drew.
All right.
Oh, there's Jim. Jim.
This is for Zoe. You mentioned both of you mentioned the use of, laser ablation spectroscopy. And Indrani explained how she was able to use it. You described the different growth bands in bones. Has there been any indication from the ablation spectroscopy that you can pick up migration, at least north and south?
Yes, so
from different bands?
There's hundreds of studies using laser ablation, the long word, inductively coupled plasma mass spectrometry, and it's predominantly used on fish ear bones because they've got that beautiful banding. And we love that because we can put a timestamp on it. And even if the fish was collected 40 years ago, we can put a timestamp on it, and we can we can create chronologies over decades of how the fish has moved, how the environment's changed. But yes, definitely used it to study fish migration and even more passive movement. Does that answer your question? Yeah.
Mm-hmm. Okay, Drew.
Yeah, another question for Zoe. And so you've made a general reference to fingerprinting, and we know quite well for humans, fingerprints are very good as a form of IDing without it being a destructive exercise. And so in your particular research, is there a way to do some of this chemical fingerprinting without actually needing to kill or harm the animal?
Yeah, so much what I've talked about is destructive, so there are parts that you do have to remove, so it does kill the animal or the product. The animal's already been collected from scientific surveys or fishes, so the animal has already died. But they do apply this method to particularly threatened species where one loss of animal is a big deal. So people use things like fish scales, which the fish just naturally sheds, and they can do that for, but not. There's also, for instance, for cephalopods, so squid, cuttlefish, octopus, in terms of calcified structures, there's no real body part that you can take without killing the animal first.
Okay, I'll do the pseudo audience again. Can I ask about the Northern Hemisphere? The correlation was not as good. Was it apart because of tuna, or was it different labs, more labs with different?
We haven't investigated that part, but it's quite interesting because there's this particular latitude where it varies, but the different taxa vary as well equally. I think the Northern Hemisphere has much more complicated land masses, and it's not just, I didn't mention that the oxygen isotope fingerprint is also influenced by salinity, but salinity varies a lot less. So it can be different evaporation rates and rainfall inputs that might be around those more complex land masses in the Northern Hemisphere. Because but if you can pinpoint that particular oceanic region in the Northern Hemisphere is off the latitudinal trend, you could still use it as a unique fingerprint.
That was quite impressive. The Southern Hemisphere correlation is like...
It's just this beautiful...
it's like,
and there was even like all sorts of animals in there, and I was a bit like aghast as a colleague. We can't put them all together in one graph. I think there was giant squid in that graph, so very, very different to, say, an abalone that just spends its whole life living on a rock and in often shallow water.
Positive spin on COVID then.
Yeah, yes.
Okay, Drew, You carry on. What's the
Well,
time? Okay, yeah, one last question.
One last question. I think this is a really fantastic question to finish off. So if we could get a very quick response from both our speakers, but we've got a question online here from a teacher. And they've asked if you could please provide some advice on how to get a greater number of students, especially girls, pursuing careers in science. Maybe Zoe,
or me?
Zoe?
Yes, it is, and I think it's a little bit different depending what field. So I think in physical sciences, or in engineering, it's harder to get women and girls started at the earlier ages. In biological sciences, we have, I'm more familiar with the university level than the high school level. We have more women in the levels, but they sort of drop away as we get more senior. So I think the different fields might need different approaches, but I think having active role models out there helps, but it is starting to get more women, more visible in the spotlight. And I think a lot of people don't think, when I've done lots of teaching on science communication and writing, I get people come up to me thinking, I didn't realise science was a creative discipline. And it's very creative, it's all problem solving focused, and even giving talks like today is a very creative process. So I think that somehow the way we train to write and read can be quite objective and quite factual. And I think if people realise more that it's a creative discipline and all sorts of different people, you don't have to be brilliant. You know, I think there's stereotypes that you think you might have to be brilliant in X, Y, and Z. And you could be, all sorts of diverse thinkers can be brilliant scientists. So I feel that they have to sort of fit a certain pigeonhole. And that's part of our cultural stereotypes that we've sort of, that's been held over the decades.
Indrani?
I will say, based on my own experience as to why I took up geology, and it was because of exceptional teaching of my teachers at the university, and particularly one. And I think, you know, Zoe did touch upon it, but for me what was really important was that the beauty of the subject was so clearly explained to me that you couldn't help being fascinated. And I think that's what's really important. We, you know, of course young girls need to be, and especially coming from India, I feel very strongly about it. But I think what we can just step back and just go, boys, girls, gender diverse people, doesn't matter. Let's just focus on just showcasing the beauty of the subject. You know, I didn't have stories like I always loved rocks when I was a kid. I didn't even know. Everything was the same to me, like in terms of rocks. But it was just his exceptional teaching skills that sparked my interest. And you know, there's so much to learn from that, and I think for me that was so important, that he opened a whole new world for me about what was possible using geology, and I'm sure it's the same with every other subject, science or not. And so yeah, if one can just beautifully articulate the subject and its benefit, I think students ought to be, you know, they ought to be fascinated.
Yeah, thank you, Indrani. That is right as well, and I agree. If I can put my 1 cent word, is I was hugely influenced by my mathematics teacher. She was brilliant, and my physics teacher, he was brilliant, and that was from year 10, 11. And that is, so teachers play a huge role, and I do wish that is acknowledged in the country. And the other part is role models, as Zoe said, and I hope, though it wasn't by intention that both speakers were women, they were just the two best scientists and best communicators which matched this series. And I guess this question has arisen because both are women, which is an interesting aspect. But it's the role models do matter. And one of the places where I had given a talk, and by majority, the response was, but she was normal. and simply because in the newspapers or something, it is some weird-looking scientist.
Or you look normal. Or
Yes. Or peering through a big magnifying glass, so one eye looks big. And whenever I see those pictures, I feel very unhappy because we are normal people.
And I think it's about getting scientists and getting more out in the community and realising we are just people, and and I think once people realise that, I think sometimes our discipline's a bit hidden, and that's partly our fault in terms of people don't realise what our day-to-day job is, but it's all about communicating and getting out there.
Yes, we say that, but then I think economics never communicate very well, and they are taking the world, let's say, down the gurgler, perhaps. But okay, I think maybe I shouldn't make any political statements. Anyway, I think we are well past 7 o'clock, but it was an excellent discussion, and thank you very much, Zoe, for coming physically to Canberra, and I'm sorry we didn't put out a more beautiful day for you. And thank you, Indrani, for staying up so late. I really, really appreciate that, and we look forward to you coming back to Australia, which you will be soon. Thank you to the audience for their questions, and the online audience as well. Thank you so much for the great questions. And thank you to my co-convener, Drew, for being there. And we would like to thank Harbour Catering and Jira at Gia Creek Station Wines for refreshments earlier this evening. And thank you all for joining us tonight. And we hope to see you again for the next installment of Surprising Science on 9th of August. And thank you, and a very good night to everyone.